we'll be focusing on biosimilar medications. I know there's a lot to discuss in terms of biosimilars. It's a really hot topic in pharmacy right now. But just to make sure we distill it down to a feasible amount of information, the presentation today will be a high-level overview on practical pearls for the practicing pharmacist. I'd also like to send a huge thank you and shout out to Philip for serving as my content expert and helping me present this or create this presentation for you all today. So just to briefly summarize what we will be going over today, we'll start with a lot of key definitions and an overview on biologics. And then we will discuss switching between products, medication safety considerations, as well as briefly touching on market impacts of biosimilars. There are 3 key learning objectives for today's presentation, and we'll be completing a knowledge check corresponding to each of them throughout the presentation. So at the end of the presentation, I hope that everyone is able to distinguish the difference between a biosimilar and a generic drug product in both the composition as well as their approval pathways, to demonstrate how to use the Purple Book and identify what information it can provide about biological products, and then determine reasons to and reasons not to switch between biosimilar products and review what should be monitored when patients do have to switch between products. So without further ado, let's begin with an overview of biologic medications. So to kick things off, I want to discuss what the definition of a biological product is. A biological product is a product characterized by both its size and its complexity. These products, unlike small molecule drugs, the majority of drugs on market are derived from living organisms rather than being produced synthetically in large batches. Our most common biological products are in therapeutic proteins, monoclonal antibodies, vaccines, and agents used for gene therapy. This is not a comprehensive list of all biological products, though. On the right hand of this slide, there's an image that depicts just how large these products are when compared to the small molecule drugs. Aspirin, which is on the far left, represents a small molecule drug for comparison. And then the following 3 on the right, the insulin, erythropoietin, and the IgG are all biological products. So you can see that even within the biological products, there's a wide range of size and complexity for these agents. A small molecule drug does not contain any peptides greater than or equal to 40 amino acids in length. So therefore, once you've surpassed that 40, you are considered to be a biological product. So once you have an understanding of what a biological product is, the next 2 definitions that are key to mastering are reference product and then biosimilar. A reference product is a biologic product that has gone through the approval process with the FDA and is now being mimicked by another drug producer to make a biosimilar. The reference drug is the drug that biosimilars are compared to, the original product. A biosimilar, on the other hand, is the product that is highly similar to and has no clinically meaningful difference from an existing reference product. The key part here is that it is a highly similar product and not an identical product. And it's expected that it will not be 100% the same structurally as the reference product. The approval pathway for these 2 classes is very different, as we will see in just a few slides, the second section of this presentation. The image on this slide shows where the difference in the 2 products are, as outlined by the bracket on the right product. Despite this minor difference, though, the biosimilar product will still be clinically as effective as the reference product. So because they're so large and complex, these small little differences make no clinically meaningful difference. So these small subtle differences between biologic products is termed inherent variation. Inherent variation is the small differences between these products as a result of the manufacturing process. As depicted in this slide, inherent variation can take many forms, and it could be one or multiple, including disulfide shuffling, pyroglutamate, deamidation, exation, fragmentation, and glycosylation and truncation. Those are all things I'm sure we learned in biochemistry back in the day, but those small little differences can be ultimately what makes the difference between a biosimilar and a reference product. It's also important to note, though, that inherent variation not only exists between the reference product and the biosimilar, but it also exists between 2 lots of the same product, meaning that even within a reference product, we see variation. So patients who have been stable on a reference product for a long time are going to have many lots of the medication that they use to inject once a month or however often they're doing it. So they experience this inherent variation without even switching to a biosimilar. Then our next set of definitions is to discuss an interchangeable product as well as an unbranded product. An interchangeable product is a biosimilar product that has gone through additional testing and approval requirements to allow it to be substitutable for a reference product. Without this designation, biologic products are not able to be substituted by a pharmacist without first contacting the prescriber. This is where they really are much different than a generic medication. We can't just substitute them as we'd like. We have to get that extra approval unless they have an interchangeable status. So just because a product is not interchangeable, though, it does not mean that it works any differently necessarily than the reference product. Rather, it's that when going through the approval process, the manufacturer did not complete this additional requirements needed for the designation of interchangeability. And we'll go through further during the approval process section of what this extra stuff they need to do to get this designation. The other definition here, the unbranded product, is not necessarily a common biologic term. Rather, I'm calling it out here to eliminate confusion regarding these products for biosimilars. For some older biologic products, some like insulins, an unbranded product is the same product and the manufacturer is just as a branded manufactured product. It's simply just sold under the generic name. A good example of this will be shown later, a Humalog product sold as insulin Lispro. So it's the same identical product to Humalog, it's just labeled as the generic or the non-branded name. So these products do not need to go through any additional approval as they're the same as the branded product, and they're simply just sold under a different name and just likely for a different price as well. Next, we can discuss how biosimilar products are named, since they do have a little bit of a silly name with the original, the kind of key root and then that suffix. So a reference product has the scientific name that is used as the root for all biosimilar names. In the example on the slide here, we'll discuss Humira. Its scientific name, adalimumab, is that root that will be attached to all of the biosimilars. So when the biosimilar product is created for Humira, it will be named adalimumab plus a 4-letter suffix. That 4-letter suffix is not chosen by the manufacturer. Rather, it's assigned by the FDA. And the FDA requires that this suffix must be unique and without any meaning, and it should not spell a word or contain any proprietary information. It has to have 4 lowercase letters, and of these letters, 3 of the 4 must be unique. So there can only be 1 duplicate letter. So therefore, kind of rounding out our naming thing here, one of the Humira biosimilars that will be released soon is adalimumab BWWD. It's also important to point out, though, that biosimilar products, unlike generics, can also have a brand name. And so you don't necessarily need to call it the adalimumab BWWD. It'll likely have a brand name associated with too that'll be more catchy and whatnot for people to remember. It's just the scientific portion that's assigned to them by the FDA. So this next slide can be, I think, a little bit overwhelming. It's a lot of text there, but listed here are all of the 39 currently approved biosimilar medications, along with their 11 reference products and their approval dates. So there are not very many biosimilars out there right now, considering how many biologic medications there are, that only 11 of them really have biosimilars at this point. So we would expect this number to continue to grow every year. The products that are in the red text are the 26 currently available to purchase, while the ones in the black text are approved, but they're not yet available on the market. Additionally, I starred and bolded the ones that have achieved the interchangeable product status. There's only 3 of them, and only 2 of them are currently available to purchase. That's the Semglee, which is an insulin glargine biosimilar, and the Cimerli, which is a Lucentis biosimilar. So this means that for the majority of biological products, pharmacists are unable to substitute without the authorization of a provider or a delegation protocol. The very first biologic approved was Zarzio, which is a biosimilar product for Neupogen. As you can see, the amount of drugs approved after that picked up pretty rapidly, and there's been 6 approved in this last year alone. The disease states treated with biosimilars is also growing beyond the initial oncology and inflammatory disease space. So we'll know that there's going to be a lot of education needed for those new sets of providers that are going to be entering the biosimilar world soon. What you can also see from this visual is that after the approval date, the path to availability is not always the same either. So some products that were approved back in 2016 are still not available, while others that were approved just last year are already on the market. So with that, we've reached our first knowledge check and our definition breather. So if everyone could take a minute to answer the question on the screen by submitting your answer to the chat. Which of one of the following statements is true regarding inherent variation of biological products? All right, I'm seeing a bunch of B's come through the chat, which is the correct answer. So all biologic products are subject to inherent variation, both the reference and the biosimilar. Within a given product, you will also see that inherent variation between lots and batches from differences in all those different kind of biochemistry things we discussed earlier. So good job, everybody in the chat. So next, we'll shift away from all of those kind of foundational definitions and go through the various pathways to drug approval, highlighting both our small molecule drugs as well as our biologic products. So this slide is, again, pretty text heavy, but has a lot of good information here. It's important to distinguish the difference in pathways to reach approval, both in our generic, our brand, our biologic, and our biosimilars. We'll start with the 2 columns on the left for our small molecule drugs. A small molecule drug must either go through a new drug application, the NDA, or an abbreviated new drug application, the ANDA. An NDA is used for any new branded product to complete, and it must undergo complete safety and efficacy studies, with the number of studies that they have to complete themselves is based on their 505(b)(1) or 505(b)(2) status, which we'll get to on the next slide. But on the flip side, the ANDA is available for any already approved product, aka this is the pathway any generic drug will go through. They don't need to complete those safety and efficacy studies like the NDA, but rather just needs to provide bioequivalence to the original medication. This pathway is much faster than the NDA pathway, as you would imagine. On the right side, we have the biologic pathways. These have 2 kind of similar routes to the NDA and the ANDA. On the left is the biological license application, or the BLA, and that's similar to the NDA, but is very specific for biological products. There is a similar biosimilar license application that is abbreviated like the ANDA to focus on bioequivalence rather than the safety and efficacy of the drug. So important to point out here too, that small molecule drugs are approved using the Federal Food, Drug, and Cosmetic Act, whereas the biologic products are approved via the Public Health and Service Act. As far as timelines go for each of these, NDAs take anywhere from 10 to 15 years, biologics can take about 15 years, biosimilars take roughly 8 to 10 years, while generics take 6 months to just a few years, depending upon how complex they are. So really, a generic drug can get done in just a few years, but a biosimilar can still take 8 to 10 years, almost the same amount of time as an NDA. The amount of time and resources and effort that goes into biosimilars is much greater than a generic, just another reason why they're really not too similar after all. Then taking it one step further, we can break down that 505 pathway between parts B1 and B2. So the 505J, the ANDA, I won't touch too much on. This is just the generic approval pathway. It has its own studies, but those studies are really just trying to prove it's an identical to the active ingredient. So the 2 different NDA pathways, the 505B1 is what we typically think about in the drug development pathway, where the product completes all of its own studies. whereas the 505(b)(2) application, on the other hand, allows drug applications to use some of their own studies, as well as then to borrow studies that were not necessarily conducted by themselves that demonstrate safety and efficacy of their product. And so this would be used when a manufacturer is developing a new dosage formulation, strength, route of administration, if it's a combination product, changing a salt formulation, a dosing regimen change, routes of administration, or if there's a new indication or a new combination. So I'm sure you're wondering why I went into all this detail on this pathway, as it's usually only used for small molecule drugs. But historically, this actually was the only pathway that existed. It was in 2010 that the PSHA pathway kind of came to the site. And so some of our older biologic medications, like our insulins, were actually approved through this 505 pathway. And that's why there are some little nuances between them. Specifically, one that had kind of confused me or tripped me up when I was preparing this presentation was the two insulin glargine products, ones I'm sure we're all familiar with. Lantus was approved first, and then Basaglar came a little while later. Basaglar is not considered a biosimilar of Lantus, rather it's its own product because Basaglar went through the 505(b)(2) pathway and became its own kind of new drug entity rather than after the biosimilar pathways came along where it would have been more of a biosimilar or a new biologic. So then shown here is another visual that helps to kind of depict how the two pathways for our biologic medications are approved and the differences between them. So with our biologic, we're really trying to ensure the clinical effect for each indication, just like a new drug application. It's this new medication we're trying to prove effective, whereas our biosimilar ensures similarity really to just the reference drug. And so for our biologic medications, they're approved on this foundation of large clinical studies supported by pharmacology and preclinical trials. So multi-center trials, large patient populations, and then a short, rigorous amount of effort that's put into the analytical studies. But our biosimilars, on the other hand, aim to demonstrate through analytical studies that you have the same molecule, or this molecule that you have has the same amino acid sequence and minor differences that will still kind of produce the same results as the biologic product. There will be some clinical studies, preclinical studies, and smaller clinical studies that demonstrate its behavior, but really those are the smaller portion of the effort that goes into proving these medications, because you actually don't even need to have studies that prove effectiveness in every disease state for a biosimilar medication. Rather, you're able to just extrapolate some results from the reference drug as long as they did a good job of demonstrating that they're biosimilar. So then the last part of the approval pathway that we'll retouch on is the concept of interchangeability. As we discussed, interchangeability status requires a few extra steps that's deemed by the FDA. So per the FDA, the biological product must first be proven biosimilar, so go through the same process, but then it must also demonstrate that it can be expected to produce the same clinical results as the reference product in any given patient. And then furthermore, for a biological product that's administered more than once to an individual, so thinking about our products that are administered once a month, once every 3 months, the risk in terms of safety or diminished efficacy of alternating or switching between the use of the biological product and the reference product is not greater than the risk of using the reference product without such alternation. So these characteristics are typically satisfied all through our clinical studies. And it's that last part there of the switching between products that's kind of the extra thing that other biosimilar studies don't typically assess, is what the differences are when patients are switched back and forth between reference product and biosimilar. It's also important to note that the interchangeability status does not need to be granted with that initial application. Biologics can petition for this after they've already been approved as a biosimilar, and it'd be added on after the fact. So while we only have 3 now, companies later on could go and make their products be biosimilar or interchangeable biosimilars. So with that, we have reached knowledge check number 2, another breather here. So if everyone could answer the next question in the chat. So Matt is a community pharmacist who received a prescription for Neulasta. Upon processing the prescription through the insurance, the claim is denied, stating that the preferred products are Fufilla or Zarzio. Which of the following statements is correct? Okay, I'm seeing a few D's come in the chat, and that is the correct answer. So in looking at these agents in the purple book, which is what we'll be discussing next, or looking at that list I originally had shared, you'll find that Fufilla is a biosimilar of Neulasta, but it does not have interchangeability status. And then Zarzio is a biosimilar for Neupogen and therefore cannot be substituted for Neulasta. So the only agents that pharmacists can substitute without provider permission or a delegation protocol similar are those with interchangeability status. All right, so next we're going to take some time to step through what the purple book is and how clinical pharmacists can use it in our practice. So the FDA has compiled this virtual database containing all the information you need to know about biologic products approved by the Center for Drug Evaluation and Research, as well as other products approved by the Center for Biologics Evaluation and Research. Some of the information the purple book can help provide for you is the date that biological products are licensed, whether a biological product has been determined by the FDA to be biosimilar, or if it has interchangeable status, the date of expiration of applicable exclusivity for biological products, and then patent information for certain licensed biological products as well. In the next couple slides, I'll have just a few screenshots from the purple book. So when you enter the purple book, there will be a search bar for you that you can search any type of biologic name. For the first example here, I had searched for Humira. When you search that, it pops up with a screen that you can scroll through that will show you what the reference products for Humira are, which products have achieved interchangeability status, and then which medications are biosimilars. One thing the purple book does not provide you with is which medications are currently available to purchase. So that's something you'd have to look up in either LexiComp or looking at your drug distributor or wholesaler database. So when looking at Humira, there's just one reference product, and then there's one product that is interchangeable, and there'll be 6 other products that are considered biosimilars. The other thing that you can see on the purple book is what different formulations the medication comes in, and so we can see Humira comes as a vial, as a pen, and then as a injector. And then you can see within the biosimilars which different formulations they have as well, or what devices they use. It's something I guess I haven't quite touched on yet, is that biosimilars don't necessarily need to have the same delivery device as their reference product. They can have a different device or some of the devices, and that might be a reason why patients might prefer some over the others, is if they have a specific delivery method they prefer. The next example that I'll walk through is insulin glargine. So this is one of our older biologics, so it's a little bit more unique and confusing. So with this one, we see that there are 4 different reference products. There's the Basilar, Lantus, Semglee, and Buechel. And so with those, they all have a different color associated with them. And that color helps to distinguish when you are looking at the purple book, which medications are biosimilar to which. And so we see that the insulin glargine AGLR and then the Semglee are both green, which means that they're only biosimilar and then interchangeable for the Lantus, that you are not able to use these products for Basilar or Buechel or anything else, that they're very specific to those products. Whereas on the Humira slide, everything was purple and everything was interchangeable with each other. So that's just another way that the book designates which things can be traded in for each other. So next we'll move into discussing switching between biologic products. Throughout my research on biosimilars, there's a few common themes that emerged regarding fears that providers, patients, or care teams may have when it came to switching between a reference product and a biosimilar agent. With these being a newer thing that people know less about, there's always going to be some fears. So the fears including immunogenicity. So there's this thought that there'd be an increased risk due to differences in the epitopes of biosimilars in the reference product, that there'd be increased side effects. I know some people sometimes report with generic medications that they have different side effects than with a branded product, but this is really throughout studies kind of deemed to be more nocebo effect, or that it's worse than patient-reported outcomes without actual changes in our objective parameters. And so patients kind of having this fear of switching and thinking that something's going to happen, and ultimately having those patient-reported outcomes that things aren't going as well. And then lack of efficacy, as well as disease stability, or disease instability in this case, So if something's broken, why fix it? Patients who are really stable on their current therapy might be hesitant to switch because oftentimes the diseases treated by these medications can get pretty severe and really life-changing. And so patients may be afraid that their disease is going to become unstable if they do switch medications. They may have went through many failed medication trials to get to their current stable regimen, and just mixing that up is something that they do fear. But overall, there was a systematic review that showed that between 178 studies, there was no major changes in safety or efficacy between multiple switches or between switches at all, and that many of the above fears are likely due to the provider's lack of experience or comfort with biosimilars, as well as just like the patient hesitancies, which acts as a perfect place to leverage our pharmacist knowledge about our biological products to kind of help with patients and providers when they do have to make the transition between products. So if patients are having to switch between products, what should we be monitoring? The first thing is anti-drug antibodies, and we should also be considering therapies that patients may be also on that could impact their ability to form these. So some patients who maybe are on things like methotrexate along with a biosimilar might be more or less likely to have these. And so studies have shown, and when we think about structurally how biosimilars are, we would assume and expect that anti-drug antibodies for a reference product would also be active against the biosimilar. So therefore, if patients have these ADAs for the reference product, we should not switch them to the biosimilar because they won't work as well. Next, we want to ensure that we're measuring objective parameters of the disease state activity and making sure that we kind of have a baseline assessment and then what the assessment is like after switching. There's always value in like those subjective patient-reported things, but it's important to also have these objective parameters to kind of support how the disease activity overall has been in a more unbiased way. It also could be useful to educate patients on what these labs and objective parameters are and what they mean to kind of help reassure them that things may be going smoothly if they are kind of apprehensive. And then lastly, while we know there is some degree of bias in our patient-reported outcomes, we ultimately do need to keep patients at the center of the care cycle and to listen to how they're feeling about the switch and helping them through any struggles that they may be having. So in looking at reasons to switch, there's both appropriate and inappropriate reasons. I'd say the most common reason we'll probably see patients needing to be switched is the payer-mandated switches. We have to have patients on the types of medications that their payer will pay for because they are expensive and they shouldn't be having to pay out of pocket. If patients are having to switch because of their payer or their insurance, this is referred to as a non-therapeutic switch or a non-medical switch because there's no real medical reason we're switching them. Rather, we're switching because the payer required it. It's also appropriate to switch based on our hospital formulary. At UW, we have one biosimilar that's preferred over the other biosimilars for each medication that we like to have patients on if they're not able to be on the one that their insurance on the outpatient side covers. And then patients should also switch based on device or administration preferences. As I mentioned earlier, sometimes not all the same devices are available in both the reference and the biosimilar. And so using the ones that work best for the patient is important. And then inappropriate reasons I've kind of touched on. So lack of efficacy of the reference product, you wouldn't want to switch to the biosimilar. We wouldn't expect it to be any more effective. And then if there's any anti-drug antibodies for either the reference product or a biosimilar, you should not be switching to the opposite one. So at UW, I've kind of touched on this. We do have a delegation protocol, our medication therapeutic interchange protocol that does allow pharmacists to switch between biologic products to either use the UW Health preferred biosimilar or to use the one that the patient's insurance prefers in the outpatient setting that you could find out through a test claim. And so this information is in the protocol, and then I believe it's under Appendix A, or if you do go in LexiComp, you're able to find the little chart explaining which one is our preferred product and the other biosimilars out there in the top part of LexiComp. So talking points for our pharmacists as we're helping our providers and patients through the switching process is really that the approval of biosimilars requires a lot of evidence of no clinically meaningful difference from the reference product. And if they do have that interchangeability status, that's even more evidence that's gone in to show that these products are safe to be switched between. There's a lot of analytical studies demonstrating that they're highly similar to our reference product. They go through animal studies to assess toxicity just like any other drug. Then there are clinical studies to show that they're safe and effective. And again, thinking back to our timeline, a biosimilar takes 8 to 10 years to approve it. Unlike a generic medication, patients usually aren't very hesitant to switch generics, and those are approved much quicker. So there is a lot of time and effort that goes into the biosimilar process. So we are now on to our third knowledge check. So if everyone could answer again in the chat, which of the following is an inappropriate reason to switch a prescription for a reference product to a biosimilar agent? All right, so I saw a couple different answers come through, so maybe it's a little bit trickier of a question. So the inappropriate reason to switch would be C. If a patient has anti-drug antibodies for the reference product, we would expect those same antibodies to be active against the biosimilar, so it would be inappropriate to switch to the biosimilar. In this case, patients should be switched to an alternative agent. So next, we're going to briefly just discuss some medication safety considerations with biosimilar medications. So one thing that I know I've had multiple people ask me when I was preparing this presentation is how to pick the right one when they're having to choose a medication that's a biologic in Epic when ordering it, if a provider says just to go and do it for them. The short answer really is there's no special strategy besides simply taking the time to double check that the product you're selecting is the right one, given all the names are going to be very similar, having that rituximab with the suffix or no suffix as shown on the screen here. It may be helpful to look over at Lexicomp to figure out which product is the preferred product or to run test claims to see which one the patients need. But really all of the names are always going to be similar, and as we see more and more come to market, they're only going to get more alike, but it just takes an extra glance to make sure that you're selecting the right product since they are so similar. And then additionally, when physically grabbing the product in inventory, it's important to remember that with the increasing number of biosimilars and even unbranded products, that the correct product is being pulled at all times. So on the left here is that unbranded product example that I had referenced earlier, where the Humalog also has the insulin Lispro box that looks almost identical to it. And I know in the outpatient setting, I once saw that this was kind of a near miss where a patient almost got a mix between these two vials. Looking at the other one on the left, right there, though, there are a lot of distinct differences between the packaging, but we know that the labels that print off in central pharmacy usually just have that more generic name on it that'll be very similar. So taking the time to make sure that the suffix is matching up with the product that you are selecting and labeling. Since payers, if the wrong thing is administered, may not cover it, or so on and so forth. So then finally, we'll conclude today's presentation with a brief discussion on market impacts of biologic medications. I know it's been a hot topic within pharmacy given the launch of Humira coming soon here in 2023. So first, I haven't quite touched on yet the market exclusivity timeline for any biologic product. So we'll go over that now. After licensing, a biologic product maintains exclusivity for 12 years, which is quite a long time. And so that's why we don't see our biosimilars coming to market right away. It's very similar to, our NDA and ANDA kind of timeline. But then with biologics, many of them also have rare disease indications, which through the Orphan Drug Act can extend the exclusivity for that indication, not the whole thing, just that indication for an additional 7 years. And so what we'll see with biosimilars is rather than waiting for the full exclusivity to be up, the biosimilar manufacturers can launch their products with a skinny label or a label that doesn't have all of the indications for that reference product to make sure that they're not violating the exclusivity time period. And then once that exclusivity is up, then they can add those indications on. So also being careful with all these biosimilars coming out that we know which indications they are approved for, that our patients are getting the right product in that regard. Additionally, similar to incentives for the first generic, the first biosimilar is granted a 6-month exclusivity period. This means that we wouldn't expect the first biosimilar to be priced much lower, and based on what we discussed in the next slide, payers may not really be incentivized to switch patients over right away to the biosimilar medication because it will be a little bit more expensive. So with Humira, with one of them going live in January, we'd expect that one to not really have too much of an impact. But then after 6 more are released in June, that's when we'll see prices drop and maybe more of the market will be impacted. Thinking about payer considerations, ultimately the one who typically decides which product we're dispensing and which one the patient received is the one who pays for it. And so we see biosimilars are coming to market. The reference product manufacturer will often offer payers rebates as a way to maintain preferred status in their formulary. So they're going to pay those payers to keep, or to those discounts to keep their product as a preferred status so patients still get it over the biosimilars. We also know that there's kind of some generic substitution laws. They vary by state, but those do not apply to biosimilars. So really the only one who determines whether a patient gets a biosimilar or not is going to be who does pay for it in the end. And briefly here, just to wrap up, just looking at cost savings, with more and more biosimilars coming to the market, we expect the cost savings to really keep rising. I know Humira will probably have a really big impact, maybe not right away, but down the line. So just looking here at this report, they showed how much biosimilars have started to save us already. So 2010, when that law was first enacted for that biosimilar approval pathway, we saw about $20 billion saved. And it was a very similar amount in the 2015 to 2019. But now that biosimilars are really starting to boom from 2020 to 2024, we expect over $100 billion of savings because of these medications. And so that number, I'm assuming, will keep going up. I'm not too savvy on this kind of stuff yet, but that'll be exciting to see in the future to see how this impacts overall the cost to our patients and whatnot. So with that, I will conclude my presentation for today, and I would take any questions that you guys have. I also have my references listed here and the CE code again for you guys to text in.